You know what's amazing? Everything about you, your looks, your personality is all stored in this little thing called DNA. Our body has 37.2 trillion microscopic cells, and each one is coded to do a different function. But what is responsible for giving out these instructions? And why do some people have black hair and hazel eyes and blue eyes and blonde hair? None other than DNA. It makes us, us. Stay till the end of the video as we will extract real DNA from our strawberry and see it with our bare eyes. Study with Ruslana.
Ruslana science!
DNA is found in almost every single cell and every living thing in this world. It is located in the nucleus, which is like the brain of the cell. It has a massive role. It contains the instructions needed for an organism to survive and develop. But just like most science terminology, DNA is only the abbreviated form of deoxyribonucleic acid. All of our traits are coded in the DNA, and we inherit it from our parents. It determines characteristics like eye color, hair color, or even if we are at risk at certain diseases. And that entire DNA code is found in all of our cells. DNA is a nucleic acid, which is a type of biomolecule. It has two strands which are twisted, and it is what scientists call a double helix shape. The structure of DNA consists of nucleotides, and there are three main types: deoxyribose, which is a sugar, and phosphate. And these hold the most important part of DNA, the nitrogenous bases. And there are different types of bases, and it is a sequence of these bases that actually determine different characteristics: adenine, thymine, cytosine, and guanine. These four bases actually pair in a specific way: adenine with thymine and cytosine with guanine. But then again, science terminology is difficult to remember, so we will use this popular mnemonic to know what pairs with what. Apples on a tree for adenine with thymine, and car in the garage for cytosine with guanine. It is the sequence and amount of these bases that actually determine if you have blue or green eyes, or black or red hair. The two bases pair in the middle of the two strands and are held together by hydrogen bonds. But is DNA just scattered around in our cells? Nope. It is organized in chromosomes, and we have 46 chromosomes in every cell, and they have a protein structure. Genes, on the other hand, are portions of the DNA strand that code for protein in a process called photosynthesis. So they would create the protein involved in producing skin color pigment, for example. But they have other vital roles too, like protecting the body, acting as transport, or making structures. However, genes in certain areas of our body can be switched on and switched off, depending on which gene needs to be used, like where this gene is relevant. And this is called gene regulation, which is done with the help of another protein called the regulatory protein. Yes, protein is very important in our bodies. Back to the gene regulation topic. Some can have a negative transcription factor, deactivating a gene, or a positive transcription factor, activating a gene. Some people study gene regulation as a career, as this quite complex topic can help us curing with future medical diseases. Our body is constantly producing new cells. Wait, does this mean it needs to create new copies of DNA too? Yes, in a process known as DNA replication, which is the same for prokaryotic and eukaryotic cells. And once again, DNA replication is done with the help of more proteins. They are called enzymes, and they have the ability of constructing or breaking down molecules. There are many enzymes that are involved in DNA replication, but we will discuss only the most important ones. Helicase. This enzyme helps to separate the two DNA strands by breaking down the hydrogen bonds and turning the DNA into RNA, which is only single-stranded. Polymerase. This helps to duplicate the DNA molecules to recreate the second DNA strand to the already existing RNA strands to turn it into DNA. But the polymerase wouldn't work without the primase, which indicates where the polymerase should start building. So let's see all these enzymes in action. Well, not literally. DNA replication will start at the origin. Eukaryotes have multiple origins, whereas prokaryotes only have one. That is where the helicase will come in and will start unwinding the two DNA strands. And the single-stranded binding proteins will make sure that the two strands won't come back together. Then the primase comes in, produces the primer, and the polymerase begins reconstructing the second strand. And that is the catch. The two strands, they're not identical. As one strand will be the leading strand, the polymerase will be moving forward as the helicase will continue unwinding the two DNA strands. Meanwhile, the other polymerase will be creating the lagging strand, as it will be moving backwards and will have to come back to the front. However, the lagging strand will end up with gaps, so let's call the ligase enzyme to the rescue. The ligase enzyme glues the gaps and completes the two strands. And here we have it, a new duplicated DNA strands. They are semi-conservative, as there is a strand from the original DNA molecule and the new duplicated strand. And this process of DNA replication is probably happening inside of you right now. Cells are literally the building blocks of life. However, there are two major kinds that you would have heard I mentioned before. Eukaryotes. These include fungi, plants, and animal cells that have a nucleus. And prokaryotes. They don't have a nucleus and they involve bacteria. But both prokaryotes and eukaryotes have DNA. It's just that we find it in the nucleus for plants, animals, and fungi, and in the cytoplasm for bacteria. Yes, even bacteria has DNA. Before we move on, I'd like to give a special mention to Rosalind Franklin, who was the first person to discover how does the DNA molecule look like. However, her work was stolen and she was only recognized for it after her death. But today marks the platinum jubilee of the discovery of DNA. Yes, on April 26, 2023, the discovery of DNA turned 70 years old. Sure, DNA is microscopic, but if we were to stretch out the DNA in one human cell, it would be nearly 2 meters long. And if we were to stretch out the DNA in all of our body, it would go from the Earth to the Sun and back 600 times. There are around 250 million bases in a single chromosome. However, humans have lost over 500 DNA codes throughout evolution. Not only that, but did you know that we shared 40 to 50% DNA with cabbage? DNA is truly amazing, but we don't actually need a microscope or a laboratory to witness it. In fact, we can see the DNA from a human right now. Well, we might need a microscope to do that, but we can extract the DNA from a strawberry right here. Let me show you how to do this experiment.
I want to see DNA.
Are you ready?
Yes.
Let's start.
Yeah.
We're going to take 1/3 of a cup of water and pour it into a container. Next, we will use half a tablespoon of salt, and we're going to add a tablespoon of washing liquid. This is our extraction liquid that we will use to extract the DNA from the strawberries. We're going to set this aside. Now we're going to prepare a tall glass with the funnel inside. And we'll either use a sieve or a cheesecloth. We're going to set this aside as we will use it later. Now let's begin with the strawberries. We're going to start with a Ziploc bag. And we're going to put around 2 to 3 strawberries inside the bag after discarding the green leaves. We will put the strawberries inside, remove all the extra air, and seal the bag tightly. Now we're going to smash the strawberries for our DNA. After 2 to 3 minutes, it will come out looking like this. And that is when we will take our extraction liquid that we have prepared earlier, and we are going to pour it into the bag. Take the spoon out. And we're going to pour this whole thing inside. Add in the extra spoon. Push out all the air and reseal the bag tightly. We will notice that the bubbles have started to appear because we've added the detergent. The detergent actually helps to lyse or pop open the strawberry cells so that the DNA could come out. and the salt creates an environment where the DNA could separate apart. Now we'll have to get ready to smash the strawberries again.
I will too.
Now let's do the fun part. We're going to take our bag and gently pour the strawberry mixture through the sieve. Can you hold this, please?
Yep.
Hold it really tightly. Let's move the sieve. And the strawberry mixture would go through the funnel, and only the clumps will be left. Now the moment of truth. We're going to take this rubbing alcohol, any alcohol of 70% will work, and we're going to gently pour it down the side.
Wow, it's amazing.
We're going to keep pouring it until it formed a 1-inch layer above the strawberries. But remember, don't let the strawberry mixture and the alcohol mix.
What happened now? DNA.
Do you see these white strings? This is the DNA. A strawberry actually has 8 genomes, whereas normal plant cells only have 1, making it perfect for this experiment, as we can see the DNA more clear. Whether you are a human, dog, tomato, or bacterium, every single cell in your body will have DNA. Knowing how to extract DNA allows scientists to figure out new medicine, know who's the suspect of the crime, genetically modify food, or even bring back evolutionary species. In Britain, scientists are developing a technology that allows us to create new medicine based on the genetic mutation of a person. Even common foods like corn, potatoes, and bananas were genetically modified to look the way they look today. Police now use clues like fingerprints or even strands of hair to find out who was there in the crime scene. This was first used in 1986. Did you know that recently, scientists in Australia managed to recreate mammoth meat? These species are already extinct. However, they use their sequences of their DNA to make this possible. DNA makes the most impossible of ideas possible. DNA is the key to our survival. DNA is crucial. So next time you look in the mirror, remember everything that makes you the way you are, your strengths, your weaknesses, your quirks, is all encoded in your DNA. And that's something truly worth marveling at. That was Ruslana Science. I'll see you in my next video.